Fatty acids, these tiny molecules are at the center of some of the most crucial biochemical processes in our body. But how well do we really understand them? Have you ever wondered how these compounds fuel your body, build critical structures, and even influence major diseases like diabetes or heart conditions? Today, we're diving deep into the fascinating world of fatty acids. Whether you're preparing for your ASCP MLS or MLT exam, exploring biochemistry in medical school, or simply brushing up on your medical knowledge, this video is tailored just for you. We'll break it all down, from the basic structure of fatty acids and their classifications to the intricate processes of metabolism, energy production, and their clinical significance. By the end of this video, you'll have a clear understanding of why fatty acids aren't just a part of your diet but a cornerstone of life itself. So, buckle up, because we're about to make fatty acids easy, engaging, and unforgettable. Let's start with the basics. What exactly are fatty acids? At their core, fatty acids are molecules with a general formula of RCOOH. The R represents an alkyl chain of carbon and hydrogen atoms, while the COOH is the functional group that gives them their acidic properties. This structure forms the foundation for their wide-ranging roles in biology. Now, fatty acids come in different chain lengths, which determines how they function. Short-chain fatty acids, with 2 to 4 carbons, are typically found in the gut, where they play a key role in gut health. Medium-chain fatty acids, which range from 6 to 12 carbons, are easily absorbed and metabolized, making them a popular energy source in clinical settings. Long-chain fatty acids, with 14 to 26 carbons, are the most common and are crucial for human nutrition and metabolism. When it comes to humans, the chains typically have an even number of carbon atoms. But there's more. Fatty acids can also be classified based on their saturation. Saturated fatty acids have no double bonds, meaning their carbon chains are fully saturated with hydrogen. These fats are often solid at room temperature, like butter. Monounsaturated fatty acids, on the other hand, have one double bond. A good example is oleic acid, found in olive oil. Then, we have polyunsaturated fatty acids, which contain multiple double bonds. Think omega-3 and omega-6 from fish oils. Interestingly, the double bonds in polyunsaturated fatty acids are spaced 3 carbons apart, a structural feature that's essential to their function. To make things even more precise, scientists use numbering systems to describe fatty acids. The delta system starts counting from the carboxyl group, while the omega system starts from the methyl end. For instance, linoleic acid, an essential fatty acid, can be represented as C18-2N6 or C18-2-delta-9-12, showing its chain length, double bond count, and bond positions. And let's not forget about the shapes of fatty acids. Cis fatty acids, which are naturally found in mammals, have a 30 degrees bend in their chain, preventing tight packing. This is why olive oil and other plant oils remain liquid at room temperature. Meanwhile, trans fatty acids, often produced through food processing, have a straight structure similar to saturated fats. Unfortunately, trans fats are associated with an increased risk of coronary heart disease, prompting efforts to minimize their use. Now that we've established the basics, let's dive deeper into their dietary significance. Let's talk about where they come from and why some are essential for human health. Fats make up roughly 40% of a typical Western diet, and about 90% of those fats are fatty acids in the form of triglycerides, phospholipids, or cholesterol esters. These are not just energy sources, they also play critical roles in cellular structure and function. Among these fatty acids, some are classified as essential. But what does that mean? Essential fatty acids are those our body cannot synthesize and must be obtained through our diet. The two main types are linoleic acid, an omega-6 fatty acid, and alpha-linolenic acid, an omega-3 fatty acid. Linoleic acid, represented as C18-2N6, is a precursor to arachidonic acid, which the body uses to produce prostaglandins. These compounds are involved in vital processes like inflammation, blood clotting, and even the myelination of neurons. Without linoleic acid, these processes can be severely disrupted. Alpha-linolenic acid, or ALA, is equally crucial. Represented as C18-3N3, it serves as the foundation for longer-chain omega-3 like EPA and DHA. These are vital for brain function, vision, and reducing inflammation. ALA is commonly found in flax seeds, walnuts, and chia seeds, while marine sources like fish oil are rich in EPA and DHA. In addition to these essential fatty acids, our diet includes a variety of other fats. Saturated fats are abundant in animal products like butter and cheese, while monounsaturated fats are plentiful in plant oils, such as olive and avocado oil. Polyunsaturated fats, particularly omega-3 and omega-6 fatty acids, are found in nuts, seeds, and fish. But here's something to remember: the balance between omega-3 and omega-6 fatty acids in your diet is critical. A typical Western diet is often skewed towards omega-6, found in processed vegetable oils, leading to an imbalance that can promote inflammation. This is why incorporating more omega-3 rich foods, like fatty fish, can significantly improve overall health. Now that we've explored dietary sources, let's shift gears and examine how fatty acids are metabolized and transported within the body. Let's delve into how they are metabolized and transported within the body. Once fatty acids are ingested, they undergo several transformations to become available for cellular use. In the bloodstream, most fatty acids are not found freely circulating. Instead, they are esterified and stored as triglycerides, phospholipids, or cholesterol esters. When needed, free fatty acids are released into the plasma and transported by albumin. Remarkably, one albumin molecule can carry up to 20 fatty acid molecules at a time, making it a critical player in fatty acid transport. The metabolism of fatty acids primarily occurs in the mitochondria of cells through a process known as beta oxidation. Here's how it works: fatty acids are broken down systematically, 2 carbon atoms at a time, starting from the carboxyl end. This process produces acetyl-CoA, a molecule central to energy production. Let's take palmitic acid, a common long-chain saturated fatty acid, as an example. It contains 16 carbon atoms. During beta oxidation, each cycle cleaves 2 carbons, yielding a total of 8 acetyl-CoA molecules. These acetyl-CoA molecules then combine with oxaloacetate from carbohydrate metabolism to form citrate, which enters the Krebs cycle, the body's final pathway for oxidizing fats, carbohydrates, and proteins. Now, why is this process so efficient? The energy yield from fatty acids is impressive. For instance, the complete oxidation of 1 mole of palmitic acid produces approximately 129 moles of ATP, equivalent to 2,340 calories. That's more than double the energy obtained from carbohydrates. This high efficiency is one of the reasons why triglycerides are the body's preferred energy storage molecule. They're energy-dense and don't require water for storage, unlike carbohydrates. However, fatty acid metabolism isn't without its challenges. Long-chain fatty acids, particularly polyunsaturated ones, are prone to oxidation at their double bond sites, making them more vulnerable to degradation. This is why antioxidants like vitamin E play a crucial role in protecting fats from oxidative damage. Metabolism also adapts to the body's needs. For example, during exercise, fasting, or psychological stress, free fatty acids are mobilized from adipose tissue to meet the increased energy demand. The levels of free fatty acids in plasma can range from 0.3 to 1.1 millimoles per liter, depending on the body's energy requirements. Fatty acid metabolism is a fascinatingly efficient system, but it also sets the stage for other processes like ketone formation under certain conditions. Let's explore that next. When the body faces a prolonged shortage of carbohydrates, such as during fasting, starvation, or uncontrolled diabetes, it shifts gears and begins producing ketones. This process, known as ketogenesis, is the body's way of ensuring that cells, especially in the brain and muscles, still have a fuel source when glucose levels are low. Here's how it works. During carbohydrate deficiency, adipose tissue releases stored fats, which are broken down into free fatty acids. These fatty acids undergo beta-oxidation in the liver, producing large amounts of acetyl-CoA. Under normal conditions, acetyl-CoA enters the Krebs cycle. But during carbohydrate shortages, the cycle slows down due to insufficient oxaloacetate, a critical component derived from carbohydrates. As a result, the liver diverts excess acetyl-CoA to produce ketone bodies. There are three types of ketones. Acetoacetate, the primary ketone. Beta-hydroxybutyrate, the most abundant and stable ketone in the blood. Acetone, a byproduct that is exhaled and gives breath a fruity odor in ketosis. In physiological ketosis, such as during a ketogenic diet, ketones provide an efficient alternative fuel source. They are utilized by skeletal muscles, the heart, and even the brain during prolonged fasting. However, the liver itself cannot metabolize ketones due to the lack of the enzyme 3-ketoacid-CoA transferase. But ketogenesis isn't always beneficial. In pathological conditions like uncontrolled diabetes, ketone production can spiral out of control, leading to ketoacidosis. This occurs when excessive ketone accumulation causes the blood to become acidic, leading to symptoms like dehydration, nausea, and confusion. Severe cases can even result in coma if untreated. Fortunately, there are ways to reverse pathological ketosis. In starvation, reintroducing carbohydrates replenishes oxaloacetate, allowing acetyl-CoA to re-enter the Krebs cycle. In diabetic ketoacidosis, administering insulin helps restore glucose uptake, halts excessive fat breakdown, and normalizes ketone levels. It's important to differentiate this from nutritional ketosis, which is a controlled and safe metabolic state. However, even in nutritional ketosis, studies suggest that elevated ketone levels might temporarily increase LDL cholesterol, highlighting the need for regular monitoring. From ketones as a life-saving fuel to their potential risks in pathological states, their duality makes them a fascinating part of fatty acid metabolism. Next, we'll explore another group of lipid derivatives, prostaglandins, and their critical roles in the body. Prostaglandins may be lesser known than fatty acids, but their impact on the body is profound. These lipid compounds, derived from long-chain fatty acids like arachidonic acid, act as local signaling molecules with powerful effects on various biological processes. Structurally, prostaglandins are classified as C20 unsaturated fatty acids with a unique cyclopentane ring. Their classification is based on specific structural features, such as the number and position of double bonds or the presence of functional groups. For example, PGE2 is one of the most studied prostaglandins, with roles in inflammation and pain signaling. So, how are prostaglandins synthesized? It all starts with essential fatty acids like linoleic acid and alpha-linolenic acid. These precursors are released from membrane phospholipids by the enzyme phospholipase A2. From there, prostaglandins are produced through two key enzymatic pathways. 1. Cyclooxygenase pathway, COX. This produces intermediates like PGG2 and PGH2, which are converted into various prostaglandins and thromboxanes. 2. Lipoxygenase pathway. This creates hydroperoxy derivatives that lead to leukotrienes, another group of inflammatory mediators. The cyclooxygenase pathway is particularly important because it involves two isoforms. COX-1, which is always active and maintains physiological functions like gastric protection and blood flow. COX-2, which is induced during inflammation and is targeted by selective anti-inflammatory drugs like celecoxib. Now, let's discuss the functions of prostaglandins. These compounds influence a wide range of processes. Prostacyclin, PGI2, acts as a vasodilator and prevents platelet aggregation, especially in coronary arteries, protecting against clots. Thromboxane A2 does the opposite, promoting platelet aggregation and arterial smooth muscle contraction. This delicate balance is crucial for cardiovascular health. PGE2, regulates inflammation, pain sensitivity, and even fever. It's also involved in uterine contractions during childbirth. However, prostaglandins are not without medical implications. Non-steroidal anti-inflammatory drugs, NSAIDs, such as aspirin and ibuprofen, work by inhibiting COX enzymes, reducing prostaglandin synthesis. While this helps alleviate pain and inflammation, it can also lead to side effects like gastric ulcers and kidney issues due to reduced COX-1 activity. Interestingly, selective COX-2 inhibitors aim to avoid these side effects by sparing COX-1, but they may increase the risk of cardiovascular problems. This highlights the delicate interplay between different prostaglandins in maintaining health and the need for careful therapeutic strategies. Prostaglandins are short-lived and rapidly inactivated by enzymes like 15-alpha-hydroxyprostaglandin dehydrogenase. They're not stored but synthesized on demand, making their production tightly regulated. From regulating inflammation to influencing cardiovascular health, prostaglandins showcase the dynamic capabilities of fatty acid derivatives. Fatty acids are biochemical powerhouses, essential for energy production, brain function, and regulating inflammation. Whether you're a student preparing for exams or a professional looking to deepen your knowledge, understanding these molecules is key to mastering biochemistry in medical science. If you found this video helpful, please like, subscribe, and share it with your peers. Let us know your questions or topic suggestions in the comments below. Thanks for watching, and see you next time.